السلام عليكم ورحمة الله. إن شاء الله نستكمل موضوعنا Molecular-Biotechnology in Microbiology Part 2. ونستكمل هذا الموضوع نظرا للأهمية القصوى لاستخدام الميكروبس في التقنيات المختلفة لعلوم Molecular Biology and Biotechnology. وهنشوف مع بعض الاستفادة إزاي من خلال هذه الداتا المتوفرة لنا من الميكروبيولوجي، إزاي نحن نقدر نستفيد منها في مجالات الموليكولار والتكنولوجيا الحيوية. اسمحوا لي أن أنا أبدأ بس التارجت نفسه لهذا الموضوع من خلال مقدمة في دقيقة كده عن أهمية هذا الموضوع، من خلال طبعا هيبقى صوتي شوية متغير شوية بالتقنيات AI نظرا عشان بس يبقى تحسين الصوت للمستمع الكريم. فنستمع مع بعض.
Molecular biotechnology and microbiology focuses on harnessing microorganisms for diverse applications, including the production of food, chemicals, and pharmaceuticals. Integrating microbiology with advanced molecular techniques enhances microorganisms' understanding and practical use in biotechnological processes, driving progress in the medical and industrial sectors.
Molecular microbiology merges molecular biology with microbiology, unlocking the secrets of microorganisms. Classical techniques like agar plates and broth cultures help isolate and grow bacteria. Light and fluorescence microscopy visualize microbial cells, while staining methods like gram and acid-fast staining differentiate bacteria based on their cell walls. Biochemical tests, such as catalase and oxidase tests, reveal enzyme presence. Molecular techniques take it further. PCR amplifies DNA, gel electrophoresis separates DNA and proteins, and southern and northern blotting detect specific sequences in DNA and RNA. Western blotting identifies proteins, while Sanger sequencing decodes DNA, revealing genetic insights. Advanced techniques revolutionize the field. Next-generation sequencing offers rapid, cost-effective DNA and RNA analysis, while CRISPR-Cas systems enable precise gene editing in microbes. Metagenomics studies genetic material from environmental samples, and transcriptomics analyzes RNA transcripts for gene expression insights. Proteomics examines sets of proteins, and single-cell analysis reveals microbial population diversity. Bioinformatics tools analyze vast data from these techniques, aiding in genome assembly and phylogenetic studies. Fluorescence in situ hybridization detects specific DNA sequences in cells, while mass spectrometry identifies proteins and analyzes microbial metabolism. Together, these techniques form a powerful toolkit, enhancing research in microbial genetics, ecology, and biotechnology, paving the way for innovative diagnostic and therapeutic applications. Understanding microbial life is crucial for tackling challenges like antibiotic resistance and advancing biotechnology.
Now let's listen to a conversation between me and Dr. Nelly and AI voice about the following topic.
What are the similarities between plasmid DNA and chromosomal DNA in bacteria? Plasmid DNA and chromosomal DNA in bacteria share several similarities, including
1. Structure.
Double-stranded, both plasmid DNA and chromosomal DNA are typically double-stranded. Circular form, in many bacteria, both types of DNA are circular, although chromosomal DNA can also be linear in some species.
2. Composition.
Nucleotide composition. Both consist of the same basic building blocks, nucleotides, which include adenine A, thymine T, cytosine G, and guanine G.
3. Function.
Genetic information, both types of DNA carry genetic information essential for the survival and reproduction of the bacteria. Replication. Both can undergo replication to ensure genetic material is passed on during cell division.
4. Involvement in gene expression.
Transcription and translation. Both plasmid DNA and chromosomal DNA can be transcribed into mRNA and then translated into proteins.
5. Role in horizontal gene transfer.
Gene sharing. Both can participate in horizontal gene transfer processes, although plasmids are often more mobile and can be transferred between different bacterial species.
6. Presence of genes.
Gene encoding. Both types of DNA can encode genes that are crucial for various functions, including antibiotic resistance, metabolism, and virulent factors.
However, the summary of the previous similarities between DNA in both the plasmid and the chromosomes of bacteria have a distinct truths and characteristics. Their similarities in structure, composition, and functions highlight their importance in genetics of bacteria. يعني بنقول إن الحمد لله سواء البلازميدس أو الكروموسوم لهم أدوار كبيرة وهامة، خصائص مميزة في أوجه التشابه بينهم من حيث البنية التركيبية والتكوين والوظيفة، كما أن هناك تسليط الضوء على أهميتهما من حيث علم الوراثة البكتيرية. يعني في موضوعات كتيرة أو إحنا نقدر نتكلم عنها في هذا الخصوص، لكن حاولنا نلخصهم بشكل سريع إن شاء الله.
Now we will differentiate between the plasmid DNA and the chromosomal DNA of bacteria. In this topic, Dr. Nelly and her father are comparing them.
Difference between plasmid DNA and chromosomal DNA in bacteria. Plasmid's DNA, 1 extra chromosomal DNA of bacteria in multiple copies, 2 smaller in size, 3 not important for the survival of bacteria, 4 provides extra characteristics to bacteria for survival under harsh environmental conditions. 5 bacteria have variable number of circular plasmid DNA, 6 not packaged with histone proteins, 7 always double-stranded, 8 shows self-replication independently to genomic DNA replication, 9 do not contain vital genes and others may have functions, such as resistance genes against antibiotics and herbicide resistance. 10 do not contain introns.
Chromosomal DNA, 1 genomic DNA of living organisms as a single copy. 2 larger in size. 3 extremely important for the survival of bacteria. 4 provides all information for the regular well-being of bacteria. 5 only 1 chromosome in bacteria. 6 can be linear or circular. 7 packaged with like histone proteins. 8 can be single-stranded or double-stranded. 9 replicate during cell division. 10 contain all the information vital genes, such as replication and code structural and function proteins. 11 all mostly do not contain introns.
The last difference between plasmid DNA and chromosomal DNA in bacteria is represented by genetic transfer, at which it can be transferred only through the horizontal conjugation gene transfer between the same or different species. يعني هنا هيبقى العملية هتتم ما بين عملية التزاوج ما بين التو cells سواء إذا كانوا from the same species or different. at which there is two cells. Firstly, one which gives the plasmid called the donor, and which receives this plasmid is called a recipient. مستقبل ومعطي ومستقبل. وبالتالي هنا بيتم العملية دي عن طريق البيلس، واللي أحد الزوائد من اللي بتخرج من البكتيريا الأولانية، وبيخرج منها البلازما. بس هنا في النوع ده بيكون البلازميد اللي هو الاف فاكتور، والاف فاكتور هنا ممثل بانه الفيرتيليتي تايب اوف بلازميد اللي احنا كنا اتكلمنا عنه في انواع البلازميد، وقلنا ان ده بيبقى مسؤول انه بيبقى كارينج للجيناتيك ماتيريالز والكاركترستكس الخاصه بالبلازميد دي، سواء في انتي بايوتيك جينز او غيره. ودي بتنتقل من السبيشيز الاول للتاني، وبالتالي او من الفرد الاول الانديفيدوال الاول للتاني، وبالتالي بتنقل الصفه معاه من خليه لخليه، وبالتالي تحافظ على ان يفضل الكولوني كلها تحمل ذا سيم كاركترستكس، سواء اللي هي الاديشنال كاركترستكس على وجه الخصوص من الصفات اللي اضيفت لها هذه المستعمره. الفكره ان بتبقى بيساعد في هذا وجود انزيم اللي هو دي ان اي بوليمريز، اللي بيقوم بعمليه تكوين صفه جديده، من كمان بيطرح صفه جديده من ان هو يحافظ يعني في اضافه جديده، لانه بيحافظ على ان الجينز الخاصه بالفيرتيليتي انها لما تنتقل تحمل صفات اللي هي كابي منها وتروح للخليه اللي هي ريسيبيانت. وبالتالي يفضل عندي الكولوني having the same characteristics of like that in the old donor in the new donor. Whilst in bacterial chromosome, the genetic transfer it occurs only through cell division by binary fission within the same species. باينري فيشن في بيبقى ساعات بعض الخلط كده انه يعتبر ميتوزيس، وده خطأ كبير. باينري فيشن is classified as amitosis which known as karyostenosis, at which this type of amitosis binary fission is a mode of asexual cell division occurred in the prokaryotes by يعني special mode of this division يعني. إنما الميتوزيس is produced by spindle formation، لكن في حالة الإيميتوزيس daughter cells are produced without spindle formation. بتتم عن طريق عملية الدوبليكيشن للجيناتيك ماتيريال as shown. prokaryotic chromatin duplicated their chromosome, so this continued growth of the cell without separation until this genetic material duplicated finally producing two cells, each one have a copy of this genetic material. So this type of classification of this reproduction is called binary fission, not mitosis, as mentioned in some books, especially in the يعني بعض المراحل المدرسية أنا بلاحظ فيها يعني للأسف أخطاء كثيرة، ودي من ضمن الأخطاء. إنه يقول لك النوع الباينري فيجن أو الانقسام الثنائي البسيط يعتبر من أنواع الانقسام الميتوزيس، وهذا خطأ. زي ما قلنا حضرتك اسمه ايميتوزيس. الايميتوزيس وعلى فكرة في أنواع أخرى كثيرة غير الميتوزيس والميوزيس زي ما احنا متعودين، لكن الأنواع الأخرى دي أنا بفرد لها فيديو خاص بيها قوي، وأنواعها كثيرة قوي أو كما اللي بتحدث في حالة الليفر سيلز وغيرها، وكلها حاجات طبيعية مش بتخش تبع الكروموسومال اباريشنز أو البوليبلويدي وما يسمى ذلك، لكن ليها انقسام يخصها بأنواع مختلفة جدا في الكائنات الحية، وبما فيهم حتى زي ما قلت حضرتك الليفر سيلز اللي موجودة في اليوكاريوتيك سيلز as in human يعني.
And the bacterial chromosome/or DNA is one long single molecule of double/or single-stranded helical supercoiled DNA. In most bacteria, the two ends of the double-stranded DNA covalently bond together to form both a physical and genetic circle. The chromosome, along with several proteins and RNA molecules in the cytoplasm, forms a irregularly shaped structure called a nuclear membrane.
A prokaryotic cell typically contains only a single chromosome. Transcription and translation are typically coupled in prokaryotes. Within the nucleoid, because the chromosome contains only one copy of each gene, prokaryotes are haploid.
Chromosomal DNA contains all genetic information necessary for the survival and well-being of the bacteria. Genetic information passes from parent to new cells via the replication of chromosomal DNA. It duplicates during cell division by binary fission. Furthermore, chromosomal DNA contains introns
Note
as
that
well
the
as
majority of
exons.
prokaryotic, viral, and organelle genes are coding. These DNA are also tightly packed with histone-like proteins. However, linear chromosomes have been found in gram-positive Borrelia and Streptomyces specified, and one linear and one circular chromosome is present in the gram-negative bacterium, Agrobacterium tumefaciens.
Prokaryotic chromosome structure, DNA domains with KB/loop observed under EM. The ends of loops are constrained by binding to a structure which probably consists of proteins attached to part of the cell membrane. This organization allows the DNA to remain compact while still being accessible for transcription and replication. Additionally, supercoiling plays a critical role in maintaining the structural integrity of these domains, facilitated by enzymes such as toposymerases. These enzymes help to introduce or relieve supercoils, ensuring that DNA remains functional under varying cellular conditions.
A bacterial genome organizes as plectonemic supercoils. Half of the supercoils are present in free form, and nucleoid-associated proteins, shown as colored spheres, restrain the remaining half.
B. In contrast, a eukaryotic genome organizes as toroidal supercoils, induced by the wrapping of DNA around histone proteins.
B. This structural difference highlights the distinct mechanisms of genome organization between prokaryotes and eukaryotes. While bacterial genomes rely on nucleoid-associated proteins to maintain supercoiling, eukaryotic genomes achieve higher-order compaction through nucleosome assembly and chromatin remodeling.
D. These variations in DNA architecture are critical for regulating gene expression, replication, and cellular processes specific to each domain.
Biotechnology of microbiology and recent techniques and applications. Biotechnology in microbiology encompasses the use of microbial organisms and their derivatives to develop products and processes that benefit humanity. Recent advancements have significantly enhanced our understanding and manipulation of microorganisms, leading to innovative applications in various fields. CRISPR-Cas9 technology, a revolutionary gene editing tool that allows for precise modifications in microbial genomes. Applications include developing genetically modified strains for increased yield and resistance to diseases in agriculture. Metagenomics, the study of genetic material recovered directly from environmental samples. Applications enable the identification of novel microbial species and their potential applications in bioremediation and bioenergy. Synthetic biology combines biology and engineering to design and construct new biological parts and systems. Applications involve the development of microbes that can produce biofuels, pharmaceuticals, and biodegradable plastics. High-throughput sequencing, rapid sequencing of DNA to analyze microbial communities. Applications provide insights into microbial diversity and functions in various ecosystems, aiding in environmental monitoring. Bioprocessing techniques methods to optimize the production of biological products using microorganisms. Applications include improved fermentation processes for the production of enzymes, antibiotics, and probiotics. Agricultural biotechnology. Use of microbial inoculants enhances soil fertility and plant growth. Biopesticides utilize microorganisms to control pests and diseases, reducing chemical pesticide use. Environmental biotechnology. Bioremediation uses microbes to clean up contaminated environments, such as oil spills and heavy metal pollution. Wastewater treatment involves microbial processes to treat and recycle wastewater, improving water quality. Industrial biotechnology. Production of biofuels through microbial fermentation processes converts biomass into renewable energy sources. Bioplastics development from microbial sources reduces environmental impact. Medical biotechnology. Vaccine development uses recombinant DNA technology to produce safer and more effective vaccines. Probiotics harness beneficial microbes to improve gut health and enhance immune response. Nature Biotechnology, a leading journal that publishes articles on the latest advancements in biotechnology. Journal of Microbiology and Biotechnology focuses on the intersection of microbiology and biotechnology, offering peer-reviewed research. Applied and Environmental Microbiology covers the application of microbiology in environmental contexts, including bioremediation and microbial ecology. Frontiers in Microbiology, an open access journal that includes research on microbial biotechnology and its applications. The integration of biotechnology and microbiology has led to significant advancements in various sectors, including agriculture, environmental science, and medicine. Ongoing research and development in these fields promise to address global challenges such as food security and environmental sustainability and public health. Biotechnology, particularly microbiology, plays a pivotal role in addressing global challenges such as food security, environmental degradation, and health crises. By harnessing microorganisms' capabilities, researchers and industries are developing innovative solutions that are sustainable and efficient. First, let's talk about CRISPR-Cas9 technology. CRISPR-Cas9 is a powerful tool for editing genomes with high precision. It utilizes a guide RNA to direct the Cas9 enzyme to specific DNA sequences, allowing for targeted modifications. Now, its applications include genetic engineering, where we create genetically modified microorganisms for enhanced traits, such as increased resistance to pathogens or improved metabolic pathways. In agriculture, it helps develop crops with improved resistance to diseases and pests by modifying the associated microbial communities in the soil. Next, we have metagenomics. This involves the direct analysis of genetic material from environmental samples, bypassing the need for culturing organisms in the lab. Its applications range from microbial diversity studies, which identify novel microbial species and their functions in ecosystems, to bioprospecting, where we discover new enzymes and metabolites with potential applications in pharmaceuticals, agriculture, and industry. Then there's synthetic biology. This interdisciplinary field combines biology, engineering, and computer science to design and construct new biological parts and systems. Applications include microbial factories, where we engineer microbes to produce complex compounds like pharmaceuticals, biofuels, and chemicals through synthetic pathways. Additionally, we can design microbes to synthesize biodegradable plastics from renewable resources, reducing reliance on fossil fuels. High-throughput sequencing is another key technique. This suite of advanced sequencing technologies allows for rapid and cost-effective sequencing of DNA, enabling comprehensive analysis of microbial communities. Applications include microbiome studies, which analyze human, animal, and environmental microbiomes to understand their roles in health, disease, and ecosystem functioning. It also aids in pathogen detection, allowing for the rapid identification of pathogens in clinical and environmental samples, enhancing public health responses. Finally, we have bioprocessing techniques. These techniques optimize the use of microorganisms in industrial processes to produce valuable products. Applications include fermentation technology, which enhances methods for producing enzymes, antibiotics, and vitamins through microbial fermentation, improving efficiency and yield. Continuous processing systems are also developed to allow for real-time monitoring and optimization of microbial cultures. Let's explore the applications of microbial biotechnology. In agricultural biotechnology, we use microbial inoculants, beneficial microbes applied to seeds or soil to promote plant growth and health. These enhance nutrient uptake, improve soil structure, and increase resistance to diseases. We also use biopesticides, which are microbial agents used to control agricultural pests and diseases. This reduces the need for chemical pesticides, promoting sustainable farming practices. In environmental biotechnology, bioremediation is a key process. It involves using microorganisms to degrade or detoxify environmental contaminants. For example, specific bacteria can metabolize hydrocarbons, breaking down oil pollutants, and certain microbes can transform toxic metals into less harmful forms. Wastewater treatment also relies on microbial processes to treat municipal and industrial wastewater, efficiently removing organic matter and nutrients, leading to cleaner water and reduced environmental impact. In industrial biotechnology, we focus on the production of biofuels. Microbial fermentation processes convert biomass into biofuels such as ethanol and biodiesel, providing renewable energy sources and reducing greenhouse gas emissions. We also develop bioplastics, which are biodegradable plastics from microbial sources. An example is polyhydroxyalkanoates, PHAs, produced by bacteria as a carbon storage mechanism. In medical biotechnology, vaccine development is a significant area. We use recombinant DNA technology to produce vaccines like those for hepatitis B and HPV, which are produced using genetically modified yeast or bacteria. Probiotics are another application. These beneficial live microorganisms confer health benefits when consumed, improving gut health, enhancing immune function, and preventing gastrointestinal diseases. The production of human insulin using bacterial plasmids is a significant advancement in biotechnology, revolutionizing diabetes treatment. Below is an overview of the technique, including the process and its implications. First, let's talk about the background. Insulin is a hormone crucial for regulating blood glucose levels. Individuals with type 1 diabetes and some with type 2 diabetes require insulin therapy due to insufficient insulin production. Recombinant DNA technology allows for the production of human insulin in bacterial systems, providing a more efficient and cost-effective method compared to extraction from animal sources. Now, the process of insulin production begins with gene cloning. The gene encoding the human insulin protein is isolated from human DNA. This gene is typically synthesized or amplified using polymerase chain reaction (PCR) techniques. The insulin gene is then inserted into a plasmid vector. Common plasmids used include PUC19 or PBR322, which contain necessary elements such as a promoter to drive expression of the insulin gene, an origin of replication to allow the plasmid to replicate within bacterial cells, and a selectable marker, usually an antibiotic resistance gene, to enable the selection of successfully transformed bacteria. Next is transformation. The recombinant plasmid is introduced into a suitable bacterial host, typically Escherichia coli, through a process called transformation. This can be achieved using methods like heat shock or electroporation. The third step is expression and cultivation. Transformed bacteria are cultured in a nutrient-rich medium under optimal conditions to promote growth and plasmid replication. Once the bacteria reach a certain density, the expression of the insulin gene is induced, often using IPTG, to trigger the production of insulin precursor proteins. Then comes protein processing. After induction, the bacterial cells are harvested by centrifugation. The insulin is produced as a precursor called proinsulin, which must be processed to form active insulin. This involves the cleavage of proinsulin to remove the C-peptide, yielding active insulin. Various purification methods, such as affinity chromatography, are used to isolate and purify the insulin from other bacterial proteins. The applications and implications are profound. The insulin produced is identical to human insulin, which significantly reduces the risk of allergic reactions compared to animal-derived insulin. Producing insulin in bacteria is more cost-effective and scalable, allowing for the widespread availability of insulin for diabetic patients. recombinant insulin has been proven safe and effective in clinical use, contributing to better management of diabetes. Recent advances include improved strains, with ongoing research focusing on developing genetically modified bacterial strains that enhance insulin yield and reduce production costs. Alternative hosts are also being explored. While E. coli is the most common host, other organisms like yeast and mammalian cells are being considered for their potential to produce insulin with post-translational modifications. In conclusion, the use of bacterial plasmids for the production of human insulin represents a landmark achievement in biotechnology. This method not only provides a reliable source of insulin for diabetic patients, but also exemplifies the power of genetic engineering in medicine. As research continues to advance, further improvements in insulin production techniques are expected, enhancing the quality of life for millions of individuals with diabetes.
Within the microscopic world of bacteria, two key players govern their genetic makeup: plasmids and bacterial chromosomes. These structures, composed of DNA, dictate everything from a bacterium's resistance to antibiotics to its ability to break down environmental pollutants. Understanding these genetic components is crucial for unlocking new frontiers in biotechnology. Plasmids, often described as independent genetic units, are small, circular DNA molecules found within bacterial cells. They replicate independently of the bacterial chromosome, carrying genes that might provide an advantage to the bacterium, such as antibiotic resistance. In contrast, the bacterial chromosome is a single, larger, circular DNA molecule that carries the essential genes necessary for the bacterium's survival and reproduction. The interplay between these two genetic entities forms the foundation of bacterial genetics. Scientists are increasingly harnessing the power of plasmids and bacterial chromosomes to develop novel solutions in medicine, agriculture, and environmental science. The study of plasmids and bacterial chromosomes is a rapidly evolving field. Scientists are continually uncovering new insights into their structure, function, and potential applications. This essay delves into the intricacies of these genetic elements, highlighting their differences, replication processes, and their profound impact on modern biotechnology. Plasmids and bacterial chromosomes, while both composed of DNA, exhibit distinct structural and genetic characteristics. These differences underpin their unique roles within the bacterial cell. Bacterial chromosomes are typically large, circular DNA molecules that contain all the essential genes required for the bacterium's survival. This genetic information is organized within a central region of the cell called the nucleoid. Unlike the nucleus found in eukaryotic cells, the nucleoid lacks a membrane, and the DNA is compacted into supercoiled structures with the help of proteins. Plasmids, on the other hand, are much smaller than bacterial chromosomes. These circular DNA molecules exist and replicate independently of the chromosome. Their genetic content often provides additional advantages to the bacterium, such as resistance to antibiotics or the ability to produce toxins. The presence of specific plasmids can vary widely among bacterial strains. Some bacteria may harbor multiple plasmids, each carrying different sets of genes, while others may lack plasmids altogether. This variability contributes to the remarkable adaptability of bacteria and their ability to thrive in diverse environments. The replication and inheritance of plasmids and bacterial chromosomes are fundamental processes that ensure the propagation of genetic information. However, these processes differ significantly between the two. Bacterial chromosomes replicate during cell division, ensuring that each daughter cell receives a complete copy of the genetic blueprint. This process, known as semi-conservative replication, involves unwinding the DNA double helix and using each strand as a template to synthesize a new complementary strand. Plasmids, in contrast, can replicate independently of the bacterial chromosome. They possess their own origin of replication, a specific DNA sequence that initiates the replication process. This independent replication allows plasmids to exist in multiple copies within a single bacterial cell, and to be transferred between bacteria through various mechanisms, including conjugation, transformation, and transduction. These unique characteristics of plasmids have made them invaluable tools in biotechnology. Scientists can engineer plasmids to carry specific genes of interest, effectively turning them into vectors for gene cloning and genetic engineering. Recent years have witnessed remarkable advancements in our understanding and manipulation of plasmids and bacterial chromosomes, opening up new avenues in biotechnology. The advent of CRISPR-Cas9 technology, a revolutionary gene editing tool, has been a game-changer in the field. CRISPR-Cas9 allows scientists to make precise changes to DNA sequences, including those within plasmids and bacterial chromosomes. This technology has revolutionized gene editing, enabling researchers to knock out genes, insert new sequences, and even edit epigenetic markers with unprecedented precision. In the realm of plasmid research, scientists are leveraging CRISPR-Cas9 to develop novel strategies to combat antibiotic resistance. For instance, engineered CRISPR systems can target antibiotic resistance genes carried on plasmids, potentially restoring antibiotic susceptibility in drug-resistant bacteria. The future of biotechnology is intricately linked to our ability to harness the power of plasmids and bacterial chromosomes. As we delve deeper into the complexities of these genetic entities, we uncover a treasure trove of possibilities for developing innovative solutions to global challenges in medicine, agriculture, and environmental science.
Hey everyone! Today I'm diving into the fascinating world of recombinant DNA technology, specifically using the restriction enzyme EcoRI. If you're a biology student, this is something you'll definitely want to get a handle on. So, what's recombinant DNA technology all about? Imagine you have a DNA molecule and you want to insert a new gene into it. To do this, you need a molecular tool that can precisely cut the DNA at specific sites. That's where restriction enzymes like EcoRI come into play. EcoRI is like molecular scissors. It recognizes a specific sequence of DNA gate TC and makes a cut between the G and the A on both strands. This leaves behind what we call sticky ends because the cut ends have overhanging single strands that can easily pair with complementary sequences. Now let's talk plasmids. These are small circular DNA molecules found in bacteria. They're great for cloning because they can carry new genes and replicate independently within bacteria. By using EcoRI, we can cut both the plasmid and the DNA fragment containing the gene we want to insert. Because EcoRI creates sticky ends, these ends can base pair with each other, allowing the new DNA fragment to insert into the plasmid. But we're not done yet. To seal the deal, we use an enzyme called ligase. Ligase acts like molecular glue, binding the DNA fragment to the plasmid permanently. The result? A recombinant DNA molecule that can be introduced into bacteria to be cloned. Once inside the bacterial cell, the plasmid replicates, making copies of the inserted gene. This is recombinant DNA technology in action, and there you have it. By using EcoRI and ligase, you can create recombinant plasmids and clone genes in bacteria. It's a powerful tool in biotechnology and research. Thanks for watching and happy
What are the classical and advanced techniques of molecular microbiology? Molecular microbiology combines molecular biology and microbiology to study microorganisms at a molecular level. These techniques are pivotal for understanding microbial physiology, pathogenesis, and ecology, as well as for developing diagnostic tools and therapeutic interventions. Here are some classical and advanced techniques used in this field. Classical techniques of molecular biology. 1. Culture techniques. Agar plates, isolation and enumeration of bacteria. Broth cultures, growth and maintenance of microbial populations. 2. Microscopy. Light microscopy, basic visualization of microbial cells. Fluorescence microscopy, visualization of specific cellular components using fluorescent dyes. 3. Staining techniques. Gram staining, differentiation of bacteria based on cell wall composition. Acid-fast staining, identification of mycobacteria. 4. Biochemical tests. Catalase test, determines the presence of catalase enzyme. Oxidase test identifies organisms that produce cytochrome oxidase. 5. Molecular techniques. A polymerase chain reaction, PCR. A method used to amplify a single copy or a few copies of a segment of DNA across several orders of magnitude, generating thousands to millions of copies of a particular DNA sequence. B. Gel electrophoresis. A technique used for separating DNA, RNA, or proteins based on size and charge, employing an electric field to move molecules through a gel matrix. C. Southern blotting. A laboratory technique used to detect a specific DNA sequence in a blood or tissue sample, involving the separation of DNA fragments by gel electrophoresis, their transfer to a membrane, and subsequent detection with a probe. D. Northern blotting. Southern blotting is used for RNA instead of DNA, allowing for the detection of specific RNA sequences within a sample. E. Western blotting. A method for detecting specific proteins in a sample of tissue homogenate or extract, combining the resolution of gel electrophoresis with the specificity of immunoassays. F. DNA sequencing, Sanger sequencing, the process of determining the precise order of nucleotides within a DNA molecule, providing insights into the genetic code and mutations. G. Restriction fragment length polymorphism, RFLP, a technique that exploits variations in DNA sequences among individuals, using restriction enzymes to cut DNA at specific sequences and analyze the resulting fragments. Advanced techniques of molecular microbiology. 1. Next generation sequencing, NGS, enables the sequencing of DNA and RNA much more quickly and cheaply than classical Sanger sequencing, allowing for the analysis of microbial communities, genomes, and transcriptomes on a large scale. So it's high throughput sequencing for comprehensive genomic analysis. 2. CRISPR-Cas systems, powerful tools for editing genomes, allowing for precise modifications to DNA sequences and studying gene function in microbes. So it's a gene editing tool for targeted modifications in microbial genomes. 3. Metagenomics, the study of genetic material recovered directly from environmental samples, bypassing the need for culturing microorganisms to understand microbial diversity and function in ecosystems. 4. Transcriptomics, the study of the complete set of RNA transcripts produced by the genome under specific circumstances or in a specific cell, providing insights into gene expression and regulation. In turn, it's the analysis of RNA transcripts to understand gene expression profiles. 5. Proteomics, the large-scale study of proteomes, which are sets of proteins produced in an organism, tissue, or biological system, helping to understand protein function, interactions, and modifications. 6. Single-cell analysis. Techniques such as single-cell RNA sequencing allow for the analysis of individual cells, revealing heterogeneity within microbial populations and insights into cellular behavior. 7. Bioinformatics and computational tools, essential for analyzing the vast amounts of data generated by advanced molecular microbiology techniques, including genome assembly, gene prediction, and phylogenetic analysis. However, computational tools and algorithms are used to analyze biological data, including genomic and proteomic data. 8. Fluorescence in situ. Hybridization, FISH. A cytogenetic technique used to detect and locate a specific DNA sequence on a chromosome, useful for studying the spatial distribution of microbial cells and their genetic material. In turn, it's a technique to detect and localize specific nucleic acid sequences within cells. 9. Mass spectrometry, used in microbiology for the identification of proteins, analysis of post-translational modifications, and metabolomics, providing detailed insights into microbial physiology and metabolism. These classical and advanced techniques collectively form a robust toolkit for molecular microbiologists, facilitating research into microbial genetics, ecology, pathogenesis, and biotechnology, and contributing to the development of new diagnostic, therapeutic, and biotechnological applications. These techniques allow researchers to explore microbial life in detail, contributing to fields such as antibiotic resistance, microbial ecology, and biotechnology.
التكنولوجيا الحيوية الميكروبية باستخدام المفاعلات الحيوية. مقدمة. المفاعلات الحيوية هي أدوات أساسية في التكنولوجيا الحيوية الميكروبية، حيث توفر بيئات خاضعة للرقابة لزراعة الكائنات الحية الدقيقة. يعرف المفاعل الحيوي بأنه وعاء مصمم خصيصا لتسهيل نمو الكائنات الحية والخلايا من خلال تطبيق المنبه الفيزيائي أو الكهربائي. إنها تسهل إنتاج المنتجات الحيوية المختلفة، بما في ذلك الإنزيمات والوقود الحيوي والمستحضرات الصيدلانية والمزيد. يستكشف هذا التقرير تطبيقات التكنولوجيا الحيوية الميكروبية باستخدام المفاعلات الحيوية، ويسلط الضوء على العمليات الرئيسية والتطورات الحديثة. لتصميم مفاعل حيوي مناسب لعملية حيوية معينة، هناك حاجة إلى دراسات مكثفة حول النظام البيولوجي، مثل نمو الخلايا والتمثيل الغذائي والتلاعب الجيني والبروتين أو التعبير عن المنتجات الأخرى لفهم متطلبات الخلايا على بيئتها الفيزيائية والكيميائية. بالإضافة إلى ذلك، من الضروري التحكم في معلمات تشغيل المفاعل الحيوي من أجل تفضيل الوظائف المرغوبة للخلايا الحية أو الإنزيمات. يجب التحكم في تركيز الأكسجين المذب ودرجة الحموضة ودرجة الحرارة والخلط ومكملات العناصر الغذائية. نظراً لوجود مجموعتين متميزتين من المعرفة، وهما البيولوجيا الجزيئية وهندسة العمليات، والمفاعل الحيوي هو جوهر العملية الحيوية، فإن هناك حاجة إلى نهج منهجي قائم على العلم لدراسة المفاعلات الحيوية، ويصبح مصطلح هندسة المفاعلات الحيوية أكثر ملائمة من مصطلح المفاعل الحيوي أو المخمر المستخدم في وقت سابق. لذلك، تم تطبيق المفاعلات الحيوية على هندسة الأنسجة، والهندسة الصيدلانية، والطاقة الحيوية، وهندسة الأغذية، والهندسة الكيميائية الحيوية. المفاعل الحيوي هو وعاء مغلق يتم فيه تحويل المواد الخام إلى منتجات معين بعد سلسلة من التفاعلات الأيضية للكائنات الحية الدقيقة. تدعم الظروف في المفاعل الحيوي العملية الطبيعية للميكروبات من خلال محاكاة بيئتها الطبيعية والحفاظ عليها لتوفير الظروف المثلى لتنمية البيئة. في كلتا الحالتين، يمكن تغذية عينات التربة الملوثة في مفاعل حيوي كمادة جافة أو ملاط. في كلتا الحالتين، يوفر استخدام المفاعل الحيوي في معالجة التربة الملوثة العديد من المزايا مقارنة بتقنيات المعالجة الحيوية الأخرى خارج الموقع. تم
Conclusion: Bioreactors play a crucial role in advancing microbial biotechnology by providing controlled environments to produce various products, from biofuels to the pharmaceuticals. The applications of microbial processes in bioreactors are extensive and continue to evolve, offering sustainable solutions to meet global challenges. Continued research and innovations in bioreactors design and operations will further enhance the efficiency and effectiveness of the microbial biotechnology. وبالتالي نقدر نلخص الموضوع بتاعنا بأن المفاعلات الحيوية لها دور حاسما في تطوير التكنولوجيا الحيوية الميكروبية من خلال توفير بيئات خاضعة والتحكم في إنتاج منتجات حيوية مختلفة، سواء ذلك من الوقود الحيوي إلى المستحضرات الصيدلانية أو غيرها. فإن تطبيقات العمليات الميكروبية في المفاعلات الحيوية بتكون واسعة النطاق وتستمر في التطور، مما يوفر حلولا مستدامة لمواجهة التحديات العالمية. إن البحث المستمر والابتكار في تصميم المفاعلات الحيوية وتشغيلها من شأنه أن يعزز كفاءة وفاعلية التكنولوجيا الحيوية الميكروبية، وما لها من أهمية قصوى.